Hello everyone. Today we will be talking about the resistance exhibited by Staphylococcus aureus. Staphylococcus aureus is one of the bacteria who has become very resistant with time. So let us first read few characters of it, that it can be isolated from dry pus after 20 to 40 days. If in hospital, proper hygiene is not being maintained and people with lesions and different kind of infections are coming and proper cleaning is not done, that dry pus falls down on the ground that can contain this bacteria that can be infectious for 20 to 40 days. So see how many days it can survive there. And it can withstand the temperature of 60 degree for 20 minutes. Its thermal death to be reported is 62 degrees centigrade for 30 minutes. Some even require 80 degrees centigrade and 60 minutes of time to be killed. Some strains of Staphylococcus aureus can be killed at 80 degree for 60 minutes when heating is done for 60 minutes. Staphylococcus aureus has a bactericidal effect by UV rays. Ultraviolet rays can kill this bacteria. Hydrogen peroxide, sodium hypochlorite, crystal violet, brilliant green, unsaturated fatty acids inhibits its growth and its colonization. And this hydrogen peroxide and sodium hypochlorite are good disinfectants also. Still today, they are effective against Staphylococcus aureus. Going into the history, why Staphylococcus aureus became the resistant bacteria, and it's resistant against penicillin antibiotic also. I'm not going into the details of discovery of penicillin, but penicillin was discovered in the beginning of 1940s. And in the mid of 90, and it became, penicillin became a miracle drug against all kinds of bacteria, and it was a miracle drug against Staphylococcus aureus also. But in the mid of 1940s only, this Staphylococcus bacteria became resistant against penicillin. So what happened and how it became resistant against penicillin was that it through mutations is it got a gene, that is Z gene, and that encodes for beta lactamase. Now beta lactamase is an enzyme which can beta lactamase is also commonly known as penicillinase. This is the enzyme which hydrolyzes beta-lactam ring of penicillin. If we go into the chemical structure of penicillin molecule, we will see a beta-lactam ring there in its chemical structure. If that ring is somehow disturbed or hydrolyzed, the structure of penicillin will not be what it is, and that chemical structure, when it will not be what it is, it will not be effective against the bacterium against which it has to be used. So it was an antibiotic. Penicillin was an antibiotic which was used to treat all kind of infections, but Staphylococcus became resistant against this antibiotic in 1940s itself by developing a gene that through mutations it got a gene, that is BLAZ gene, which encodes for beta lactam, and thus it made penicillin ineffective or inactive against Staphylococcus aureus. So in early 1950s and 60s, it was reported to cause community infections and became pandemic also. S. aureus phage type 8081 was the one reported to be causing in the year when 1950s and 60s, it started to cause community infections, while in 1940s it was associated with hospital infections. In a hospital environment, it was spreading infections, but later on in the year 1960s, it started to cause community infection also. That means a person who have not visited hospital was also getting infections from this Staphylococcus aureus. So this Staphylococcus aureus in mid 1940s become penicillin resistant, And in 1961, it was reported to cause hospital-associated infections too. In between, when it became penicillin resistant in 40s, then methicillin was discovered, which added a special moiety to penicillin, a special chemical, a heavier moiety to penicillin. Methicillin is a chemical structure which is heavier than penicillin, and this methicillin was effective against Staphylococcus aureus till the time, from the time when penicillin became resistant. We started treating staphylococcus infections with methicillin. Now this methicillin moiety, which was heavier than penicillin, was effective against this Staphylococcus aureus. Later on, in 1961, penicillin-resistant organisms were also reported, and this was cases were reported in hospitals. Later on, it was community-associated cases were also reported, and these cases were reported in 1990. That means from 40s, when penicillin became ineffective, methicillin was given. Then methicillin-resistant organisms also came up, and that was hospital-associated methicillin-resistant organisms were reported in 1961, while community-associated were reported in 1990. And what was the mechanism against that was working and making Staphylococcus aureus resistant against methicillin was gene MAC-A that encodes low affinity penicillin binding protein. That means BP828. That means I told you that what is methicillin? Methicillin was a heavier moiety than penicillin. A heavier molecule was attached to penicillin, and so that beta lactamase enzyme cannot hydrolyze it. So how next this bacteria developed resistance against this new discovered methicillin? Because it got another gene, mutated gene, that was MAC-A. Now MAC-A is, now new researchers has showed that MAC-A is a chromosome which contains lot many genes on it. So that was reported, and that gene had a characteristic that it has a low affinity towards binding. That means that gene was not allowing the receptors, actually the envelope of the Staphylococcus bacteria, to receive or bind with penicillin. So it has gained low affinity to bind with penicillin binding protein, that is BPA2A. So in this case, bacteria has developed a gene, again a gene which is protecting it, which is resisting it, and making it resistant against the methicillin antibiotic. So this bacteria has become methicillin resistant, and in hospital it was reported in 1961, and in community it was reported in 1990. And the strain that is called MRSA is a strain which is associated with both hospital and community infections. And this is a methicillin resistant. M stands for methicillin resistant Staphylococcus aureus. So MRSA is methicillin resistant Staphylococcus aureus. And now this methicillin resistant Staphylococcus was resistant against broad spectrum antibiotics. That is, it was resistant against penicillin, cephalosporins, and carbamines, while penicillin resistant was not resistant to broad spectrum antibiotics. It was just penicillin resistant, but methicillin resistant become more resistant to broad spectrum antibiotics. And later on, it is very becoming difficult to treat Staphylococcus infections, which was easier when penicillin resistance was not developed, and when methicillin resistance was not developed in Staphylococcus aureus. Nowadays, vancomycin is a drug of choice. Vancomycin is a glycopeptide antibiotic, and it inhibits the cell wall biosynthesis of the bacterium. So now we still have strains which are exhibiting increased resistance to S. aureus, and these strains are called vancomycin intermediate S. aureus, that is VISA, and in which minimum inhibitory concentration is 4 to 8 microgram per ml. So concentration is less, and this is VISA. vancomycin intermediate as aureus, and that is resisting the uh, providing resistance to Staphylococcus aureus against vancomycin also. Next is complete resistance to vancomycin, and that is known as vancomycin resistant as aureus. The Visa was intermediate resistance, concentrations were low, while this vancomycin resistant organisms have minimum inhibitory concentration more than or equal to 16 microgram per ml of the antibiotics. So it is very important for us to use antibiotics properly, and each antibiotic should be given against against a bacteria against which it can work. We should not give all kinds of antibiotics without knowing the organism causing infection. This misuse of antibiotics is causing a great kind of pandemic. Thank you for patient listening.